by Lindsay Sapetti, and she worked in the laboratory of Dr. Rong Yang in our internal medicine department, and Dr. Rong asked me if I would introduce Lindsay today. So Lindsay Sapetti, our families have a history. Lindsay graduated from Rochester High School in 2020. She's now a junior at Illinois Wesleyan University studying biology. Lindsay actively participates in many social activities, including her sorority, biology club, in the financial advisory board, and student government. For her career, Lindsay plans to attend medical school, hopefully SIU, Lindsay, and have a special interest in epidemiology and oncology. It was a pleasure and pleasant experience working with Lindsay this summer. I found Lindsay to be a very quick learner, willing to work hard. In just 2 months, she mastered many cellular biology and molecular biology techniques and learned some important statistical methods and methodologies. I hope these experiences can provide a little help for Lindsay's career development. I now would like to have you listen to Lindsay's story of this summer with her gene of interest, NRIP1, in breast cancer.
Thank you, Dr. Torrey, and thank you, Dr. Yuan. So as he said, I will be presenting on the impacts of suppressing NRIP1 expression on estrogen receptor positive breast cancer cells. So breast cancer is the second leading cause of death among women. In 2020, there were 2.3 million women diagnosed with breast cancer, and it is estimated that in 2022, over 300,000 women in the United States will be diagnosed with breast cancer. So there are many different ways to classify breast cancer. When looking at the origin of the breast cancer, most breast cancers are carcinomas, which is a tumor that originates in the epithelial cells of organs or tissues. Although rare, breast cancer can also be a sarcoma, which originates in the connective tissue of the breast. So when looking at the location of the cancer, it can be ductal or lobular. So ductal is in the milk ducts, and then lobular is in the lobes, which are the glands that make milk. When looking at the status of the breast cancer, it can be invasive or in situ. So invasive cancers have spread out to other tissues, while in situ have not. So some special types of invasive cancers are listed here. Luminal A and luminal B are both estrogen receptor positive. However, luminal A is progesterone receptor positive and HER2 negative, while luminal B is progesterone receptor negative and HER2 positive. Basal-like or triple-negative cancer is just as it sounds. It is estrogen receptor negative, progesterone receptor negative, and HER2 negative. Lastly, HER2 positive cancers are obviously positive for HER2, and they're often estrogen receptor negative and progesterone receptor negative. So cancers that are hormone receptor positive have an excess for that hormone receptor, and they grow in response to that hormone. This can be important when deciding treatment options. So some treatment options are surgery, which is the removal of the cancer tissue, chemotherapy, which is using chemicals to kill the cancer cells, radiation therapy, which is the use of energy as electromagnetic waves, hormone therapy, which is blocking the cancer cells from getting the hormones that they need to grow, and immunotherapy, which is treating cancer by amplifying or suppressing the body's own immune system. So nuclear receptor interacting protein 1, also known as NRIP1, plays a role in metabolism, inflammation, as well as development and aging. It's a co-regulator for many transcription factors, such as nuclear factor NF-kappa B. So in breast cancer, NRIP1 plays a role in the regulation of the age of menarche. Earlier age of menarche is associated with the higher risk of breast cancer. Additionally, the function of estrogen receptor alpha in transcription requires NRIP1, and NRIP1 is overexpressed in breast cancer tissues and cell lines. So something else that I will be talking about is small interfering RNA, or siRNA. So this is a double-stranded non-coding segment of RNA, and gene expression can be silenced using siRNA strands with a complementary sequence. siRNA interferes before translation takes place. So it is introduced into the cells via transfection, and that is the introduction of nucleic acid into eukaryotic cells without the use of a viral vector. This can be done using lipids or electricity. So it does have some challenges. First off, siRNA silencing does not always work if the strands of the siRNA and the target mRNA are not directly complementary at the site of the cleavage. Additionally, the efficiency of siRNA knockdown can vary, and siRNA knockdown is temporary. So the goal of my project this summer was to determine the impact of NRIP1 suppression on T470 cell growth rate and on the expression of breast cancer-related genes, and this was done using the siRNA. So for my experimental design, I began by doing some preliminary tests of the siRNA knockdown efficiency, and this was used to test at which time point I would continue with. So I treated cells with siRNA to target NRIP1 and harvested them at 3 separate time points. This was at 24 hours, 48 hours, and 72 hours. So then, cells were treated with siRNA and harvested at that chosen time point, which was 72 hours. I ran 3 cell viability assays, the MTT test, cell counting, and I used the ViCell machine. So the MTT measures the reduction of a tetrazolium component by the mitochondria. The cell counting was manually counting the cells under a light microscope using a hemocytometer, and the ViCell machine measures cell viability and the number of cells per unit using by the use of trypan blue and by taking a series of images of the sample. The gene expression assay was the qRT-PCR, and I tested genes in different biological function groups, which will be seen on the next slide. So these are the genes that I tested in their respective groups. I tested genes in apoptosis, epithelial mesenchymal transition, the hormone and growth factors, inflammation, and the mTOR pathway. So here are the preliminary results. As you can see on the right-hand side, the 72-hour time point was displayed the greatest knockdown efficiency. This was about 50% knockdown of NRIP1. This was followed by the 48-hour time point, which had about a little over a 20% knockdown. So here are the relative gene expression levels at that 72-hour time point. So on the very left-hand side is the NRIP1 knockdown. I achieved a level of about a little over 38% knockdown. So most notably, there's a statistically significant decrease in the expression of the CCND1 gene. This is part of the mTOR pathway. So CCND1 is amplified in many cancers. It encodes for a protein that is required for the cell cycle, specifically in the G1 and S transition stage. So also, some of these results do not necessarily agree with previous results from our lab, so this may be due to many factors. First off, this used T470 cells, and some of the experiments in our past were using stem cells or MCF7 cells, so that is something that we'll keep in mind and would like to investigate further. These are the 3 cell proliferation assay results. So on the 3 proliferation assays showed a trend of a decreased rate of proliferation in the NRIP1 treated cells. On the bottom here is my MTT results. So my MTT results actually do not look like that of a typical MTT assay. This may be due to many things. So first off, the MTT test is actually a measure of the mitochondrial function in the cell. It's not a test of the cell number. And it runs on the assumption that the mitochondrial function of every cell is the same, when that is not always true. In fact, studies of the radiation-induced apoptosis have shown that the radiation treatment actually impacted the function of the mitochondria in cells. And then additionally, different studies actually showed that NRIP1 played a role in the mitochondrial function, and knockout NRIP1 can actually increase mitochondrial function. So I did the cell counting and the hemocytometer as well, and to verify the results of the MTT. So in conclusion, siRNA treatment can successfully knock down NRIP1 expression in T470 cells. Suppressing NRIP1 may alter the expression of other breast cancer-related genes, and it reduces T470 cell proliferations. So these findings suggest that NRIP1 may be a therapeutic target for breast cancer. So some future direction that we would like to look into is to verify the effects of knockout NRIP1 using a different mode of knockout, such as shRNA, which may last longer in the cells. Also to investigate the molecular mechanisms of suppressing NRIP1 in breast cancer, for example, to investigate the interaction between NRIP1 and the mTOR pathway, and to evaluate the level of NRIP1 expression in the days following siRNA treatment. So I would love to say a special thank you to so many people. First off, the Simmons Cancer Institute at SIU, the staff, and all of the donors. SIU School of Medicine as well, and all of the wonderful people that I've had the pleasure of meeting throughout this internship. Dr. Rong Yuan, Dr. Yun Zhu, Dia Al-Salami, and all other of the students and the lab personnel that I've had the pleasure of working with. Thank you.
Questions for Lindsay?
Yes.
So just out of curiosity, so in your RNA expression studies where you're using real-time PCR to monitor or track the amount of gene expression, was there a gene that you thought when you reduce the expression of NRIP1 that it should go up or down in response to that? Like something that it regulates directly?
Well, that was kind of the question. So it is known to be involved in many different pathways. For example, the NF-κB pathway. I had hypothesized that the NF-κB might be reduced because that is an inflammation gene, and the NF-κB actually promotes the expression of inflammatory cytokines. So that was actually something that I had found that was upregulated, which was kind of contradicted our previous results. So that was something that I would love to further investigate if I ever get the opportunity again. But pretty much all of the genes I tested were kind of hypothesized to be involved with the NRIP1 gene, but yeah.
Other questions for Lindsay? Lindsay, thank you.